Hello everyone, this is Mr. Burke, and I'm here to explain 10.1 meiosis, HL. So the essential idea for this subtopic is that meiosis leads to independent assortment of chromosomes and unique composition of alleles in daughter cells. So again, we are focusing on that genetic variation that is generated due to meiosis and due to some of the processes that happen in meiosis. So this is an image of a family, and it's just showing the large amount of genetic variation that it can occur thanks to meiosis. And why do we care about this genetic variation again? That's an important idea to be thinking about. So we're going to start with a warm-up. If you'd like, you can pause right now, try to answer these questions on your own, and then I'm going to go through the answers. Okay, so number one, crossing over happens during which phase of meiosis? If you said prophase 1, you would be correct. It does not happen in prophase 2, only prophase 1. Number 2, what is produced as a result of crossing over? If you said increased genetic variation or possibly recombinant chromosomes, you would be correct. We'll talk more about that word recombinant chromosomes in this presentation. Number 3, why is crossing over so important? So this goes back to that idea that genetic variation is a thing that we generally want in genetics and with evolution and adaptation and natural selection. As I've explained before, the more genetic variation you have, it's like having more tools in your tool belt. If an organism or a species has more genetic variation, it's considered stronger than a species that has less genetic variation. And the reason being that it increases the survival rate of the species and the organism if you have more genetic variation, because you would presumably and theoretically be able as a species to survive in a wider variety of environments and different scenarios. Does a quick review of meiosis before we get into all of the details that the higher level version does have in it. Let's just do a quick review of 3.3. One diploid nucleus divides by meiosis to produce 4 haploid nuclei. So here we have a diploid nucleus, right, over here. And what's happening right here is the chromosomes have now replicated and forming that X-shaped structure versus just the line or linear chromosomes. They're still linear, but they're copied, and so when they're copied, they have the 2 sister chromatids and do have that X-shaped structure that we talked about before. Okay, and here they are going through one phase of meiosis, because remember, meiosis goes through the 2 phases, right? They go through PMAT1 and then PMAT2. So if we go through PMAT1, then we would end up with the 2 cells, right? And then those 2 cells go through PMAT2 and they divide again to form the 4 haploid gametes, right? And remember, diploid is represented with 2N, that it represents that it's diploid, 2N. And then we've got N, which is what the gametes would be, the 4 gametes that are produced. Keep in mind, and you will run into this later with HL, that when the sperm are produced, there are 4 gametes that are produced, versus when the eggs are produced, there's 1 viable egg that ends up being produced. Yes, it still goes through all of these divisions, but in the very end of the production of eggs, there's actually only 1 that gets produced. And we'll talk more about that in future subtopics. Okay, so before we begin going through all of the details of meiosis and the important topics that the IB wants you to know in relation to meiosis, I do think it's really important to always look at these statements and the guidance statements that go along with them before you begin. So you can go ahead and pause your video, read through See which ones you would already be able to recognize and somewhat understand or fully understand from 3.3, and then try to mark the ones that maybe seem a little bit more confusing to you that you know you're going to have to focus on and review a little bit more in order to understand fully. Before we start getting into all of the little minute steps of meiosis, let's just go back to that essential idea, which is that essentially genetic variation is increased in meiosis through the independent assortment of chromosomes and unique composition of alleles in daughter cells. So what are the things in meiosis, hopefully you can remember from 3.3, that increase genetic diversity in the gametes? What are the things that increase genetic diversity? The main things that increase genetic diversity in meiosis are crossing over during prophase 1, where you have an exchange of genetic information between the non-sister chromatids of the chromosomes that line up. And then the other thing that increases genetic diversity is that random orientation of the chromosomes when they line up in the center of the nucleus during metaphase. Right, so if you go to slide number 6, you can see that there's an animation. It's really useful animation for meiosis. We're going to watch that first before we start talking about each of the steps. If you click on that link, it will download something called an SWF file. If you want to be able to play these files on your computer, you can just Google SWF player or something like that, and you'll be able to download a player like this that will be able to play it. So here we go.
In prophase 1, the DNA coils tightly and individual chromosomes become visible under the light microscope. Homologous chromosomes become closely associated in synapses, and they exchange segments by crossing over. By metaphase 1, the nuclear membrane has disappeared, and the microtubules form a spindle. Spindle fibers attach to only one side of each centromere, and the two homologous chromosomes attach to microtubules orienting from opposite poles. Each pair of homologs then lines up on the metaphase plate. Either maternal or paternal homolog may orient toward a given pole. In anaphase 1, the microtubules of the spindle fiber shorten.
Okay, so I just wanted to pause it right there because this does start to explain that random orientation of the chromosomes, as we were saying before. So if this mostly blue chromosome represents, for example, the mother, and this other mostly blue, or maybe you see it as green, chromosome represents the other chromosome from the mother, right? And then we've got the father over here with the purple, and then this purple. Basically, these can orient and arrange themselves in whichever way that they would happen randomly, okay? And it's the random orientation of them. So this blue one could be on the bottom or it could be on the top. Purple could be on the top or the bottom. And then that does influence the genes and the allele combinations of the gametes. And so it increases genetic diversity because of this random orientation of the chromosomes.
And pull the chromosomes toward the poles, taking both sister chromatids with them. Each pole ends up with a complete haploid set of chromosomes consisting of one member of the homologous pair. During telophase 1, the nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains two sister chromatids for each chromosome attached to a common centromere. Because of crossing over, the two sister chromatids are not identical. During prophase 2, the nuclear envelope breaks down and a new spindle forms. In metaphase 2, spindle fibers bind to both sides of the centromeres. During anaphase 2, the spindle fibers contract and the sister chromatids move toward opposite poles. In telophase 2, nuclear envelopes reform around the sets of daughter chromosomes.
Okay, so let's start reviewing each of the steps. We do begin with interphase because remember with interphase, this is where DNA replication is happening. Okay, and remember right here, if you see a chromosome like this with the X shape to it, it's already been what? It's already been replicated. If it's just a single one, then it hasn't been replicated yet, and they are combined with each other and attached to each other at the centromere. Okay, and more specifically, it's in S phase, right? That this replication is happening. Synthesis of DNA, S phase. Okay, so this is a karyogram and it's showing all of the chromosomes in what? Which organism do you think this is? How could we figure it out? Well, we could start counting all of the chromosomes in the karyogram and we would notice that there are 46 chromosomes. Remember that humans have 46 chromosomes. We have 23 pairs of chromosomes. So this is one pair. This is a second pair. This is a third pair right here. And then all the way to your 23rd pair, even though it's technically maybe not a pair, and that's why they're shown separately here, because it's your X chromosome right here, the longer one, and then your Y chromosome right here, which is the shorter one, the sex chromosomes, the ones that determine your gender. And so don't forget that your pairs of homologous chromosomes do come from your mother and your father. So if this is a pair of homologous chromosomes right here, you've got one of your homologous chromosomes from your father and one from your mother. And that's what I was showing too in that animation with the purple versus the green chromosome, right? One was from the father, the purple chromosome in the animation was from the father, the green one was from the mother. Okay, and now we've got after replication, this is what they look like, and you can tell they're replicated again because of that X-shaped structure. So this is a replication of the father's chromosome inside of, you can imagine, inside of yourself, and then this is the replication of the mother's chromosome inside of yourself, right? Before we go through meiosis to make the gametes, which would be egg or sperm, depending on if you're a male or a female. Okay, so this is a homologous pair of chromosomes. You can see that they do have the alleles all at the similar loci or loci, and then they're also the same length, and they have their centromeres at the same location. So those are some of the clues that you can tell that they are a homologous pair of chromosomes, one from the mother and one from the father. After they're replicated during S phase and interphase, this is what they look like. Okay, so you've got your sister chromatids, which are exact copies of each other, and then you have your centromere that connects the two. Okay, during prophase 1, something very important happens. Recombination of alleles is what it results in. The actual process of the crossing over of the non-sister chromatids is called crossing over. It's when you see those arms of the sister chromatids on the different chromosomes crossing over, right, of the homologous chromosomes crossing over and exchanging genetic information. So this is one pair of homologous chromosomes. We call them bivalents. Then we've got another pair of sister chromosomes that will also be crossing over. And again, this is when recombination of alleles takes place. That's the exchange of the alleles and the genetic information. It promotes genetic variation. So they do refer to the homologous chromosomes that are lined up with each other as bivalents. The process of them lining up with each other is called synapsis, and then the actual process of them exchanging genetic information is crossing over, leading to recombination of the alleles. There is a lot of vocab in this subtopic, so make sure that you are reviewing it. Here we've got the chiasma, okay? These are the structures that form during crossing over. So how many chiasma can you actually identify in these micrographs? If you counted 2 on this first one, you would be correct. If you counted 5 on this one, you would be correct. Okay, so crossing over does increase genetic variation by recombining the alleles. Okay, so here we have, for example, the small e allele is now being transferred to the other homologous chromosome versus this large E, capital letter E, is being transferred from this chromosome to this one.